To be able to understand how different alleles or versions of genes can combine together from different parents to produce different traits in offspring, we need to walk back and think about the first evaluation and investigation of genetics, that is, Mendel's P's. So this should help review some key concepts about inheritance and traits. First, it's important to understand the idea about how to tell the genotype of an individual and whether or not it is a pure breeding genotype, which we currently call a homozygous genotype. And the way that Mendel did this, and the way that we still do this in the laboratory, is mating two individuals together that have the same phenotype or trait. And in this case, the trait is being a yellow pea. So if we cross two individuals together and they are the same genotype, then they should produce entirely offspring that have only the parental trait. That is, all of the individuals in the next generation, without exception, have the same trait or phenotype. In this case, we will denote the version of the gene that causes yellow by a capital Y. And although you may know why it's a capital Y, we won't go into that detail yet. We're going to talk about that in just a minute. Since Mendel did these experiments, we also learned that peas are diploid species, which means they have two copies of each chromosome. So, to be a pure breeding individual means both alleles or both versions of each gene are the same, identical. So to reiterate, the way we know an individual is pure breeding is we mate two individuals with the same phenotype, or in the case of plants, sometimes because they're often self-fertile, we can take one individual and self-cross it. All of the offspring have to have the same phenotype or trait. Then that defines the parent's genotype as being homozygous or pure breeding. Mendel did exactly the same thing with peas that produced green fruits or peas, and found that all of the offspring then were green with no yellow fruits being present, even though Mendel later discovered, as we're about to see, that the yellow trait is dominant to green. So this also indicated the pea plants producing the peas that were crossed are still homozygous, but they're homozygous for the recessive allele, lowercase y in this case. And the way that Mendel determined which allele was dominant was by mating two pure breeding individuals. In this case, we've got individuals that are homozygous for the yellow producing version of the gene and homozygous for the green producing version. So these individuals in the P0 or parental generation have to be pure breeding, in other words, homozygous. The reason that's important is because that means all of the F1 individuals inherit one allele, say in the sperm of one individual, although plants don't have sperm, but that's a detail we'll talk about later perhaps, and one allele from the egg of the mother. That's the only combination of male gamete and female gamete that can be produced. So we know if we set a pure breeding cross, every single F1 individual is going to have the same genotype, which is they inherit a Y from their father, capital Y, and a lowercase y from their mother. So every single one of these individuals in the F1 generation has this genotype, capital Y over lowercase y, and then we look at their phenotype. All of the individuals are yellow. That tells us that it's the trait that came from the father, the individual that was yellow. That allele is dominant to the allele that came from the parent whose phenotype is not present in their offspring's generation. One of the experiments that Mendel did was to mate two P0 or parental generation individuals together. Again, because these are pure breeding, we know that the yellow P is homozygous capital Y, and the female P, or the green P, is lowercase y over lowercase y. This produced an F1 generation, as we just saw, with lots of individuals. Mendel chose two of those individuals, again, both of which would be yellow, and both of which would have the genotype capital Y over lowercase y, and mated them together to produce the second filial sibling, or F2 generation. And this is an example of an F2 cross design. Take two parents, mate them. In this case, siblings were mated together, that's not always the case, to produce a generation of grandchildren, the F2 generation. And in Mendel's case, what he saw was he found 6,022 yellow seed-producing or fruit-producing offspring, and 2,001 green fruit-producing F2 plants. One important thing we've learned in the over 100 years since Mendel first conducted these experiments is that it's possible to predict the genotypes of some of these F2 individuals. For example, we know that every green pea has to be homozygous for the recessive allele. Then the question becomes, what about the yellow peas? There are two different genotypes that can produce yellow peas, so we're not sure yet whether or not any of these yellow F2s might be homozygous for the dominant allele or heterozygous, because in dominance, it takes only one dominant allele to produce the dominant trait. Years later, a geneticist named Punnett discovered this simple principle that if you have two parental individuals, say again homozygous, pure breeding, for the dominant allele and the recessive allele, they can produce two offspring that are the same phenotype. That is, both of those individuals are heterozygous. They have one capital allele and one lowercase allele. And one of Punnett's contributions was this graphical representation of one of Mendel's laws of inheritance, the law of segregation, which indicates that each individual will produce half of their gametes that contain one of the two alleles they contain. The other 50% of the gametes will contain the other allele, and that's entirely to do with how meiosis works, which we'll talk about at some other point. So that's why the Punnett square is designed as a square. It has equal length and equal height divided exactly in half to represent that 50% of the gametes, let's say in the case of a mammal, if this was the female, that would be eggs. And if this was a male, that would be sperm. Exactly half of the eggs would contain the Y allele if heterozygous, and the other half would contain the lowercase y, and the same would be true of sperm from a father. And that's why we write the alleles that are present in the parent on the axes of the Punnett square. So these represent the contributions of the sperm and the eggs when they fertilize at syngamy, the combination of sperm and eggs, to produce the genotypes of the next generation, which are present inside the Punnett square. In other words, the Punnett square is a prediction of what the genotypes of the F2 generation are going to be. And as you may have seen before, you copy the sperm alleles across and the egg alleles down, and then fill in all of the squares of the Punnett square. And these represent the genotypes, or the expected genotypes at least, of the next generation, or the F2s, the children of the F1 cross. Now, importantly, there are two different pieces of information we can get out of the prediction of the F2 genotypes. One is we can get an idea about the genotype ratio, which I'll indicate with a G, and the other is an indication of what we should expect of the phenotypes of the F2 generation, which I'll indicate with the P. Now, in the Punnett square, there are three different genotypes. There's homozygous capital Y. There's one of those which produces yellow peas. And then there are two heterozygous genotypes which also produce yellow peas. And then there is the third genotype, homozygous little y, which produces green peas. And this defines the 1 to 2 to 1 genotype ratio. There are three different genotypes: homozygous capital Y, heterozygous, and homozygous lowercase y. And those are present in 25% of the offspring from a cross, 50% of offspring from a cross, and the other 25% of offspring from a cross. So with information about dominance in mind, we can then create a phenotype ratio. 3 out of these 4 genotypes have a capital Y as part of the genotype. Therefore, for every 3 individuals that are yellow, that is combining the homozygous and the heterozygous genotypes, there should be 1 green individual that has the recessive genotype that produces green peas. In other words, and this is important, if 2 F1 individuals of the same genotype, both heterozygotes, are mated, the Punnett square predicts 2 things: a 1 to 2 to 1 genotype ratio in their offspring, the next or F2 generation, and a 3 to 1 ratio of phenotypes, and that's because the dominant allele occurs and produces the same phenotype in the homozygotes as well as in the heterozygotes. So 75%, 3/4 of F2 individuals should have the F1 phenotype, and 1/4 should have the parental phenotype. And it's really interesting to geneticists when this Mendelian segregation or inheritance pattern doesn't happen.